All right. Thank you for joining for this talk, and I thank the organizers for this opportunity to share my work. So at the beginning, I just want to tell you, I'll just share a very wide sort of, just give you an overall picture of what we've been working on about living fluids. And my hope is that it'll interest you, and then if you're interested in details, we can discuss all that later. So I'll start with a brief motivation about the problem. Where does it come from? So what you're seeing here are movies of bacterial motion. These are experiments. So the first movie shows you how E. coli cells, they perform their typical run and tumble dynamics. When they are not in a very dense suspension, they're fairly dilute. So you see the cells individually. And what's interesting is that they have a velocity around 10 to 20 micrometers per second. So it's fairly sluggish. But then you just take the same cells again, you concentrate them, and you get a very dense suspension. So you have to resuspend it in some appropriate chemicals and so on. But then they sort of transition to this collective phenomena, this emergent flow, which you see on the right, which is very vigorous, looks highly chaotic. And remarkably, the velocities really go up. So this is something that the bacteria can only do in a state when they are all moving together, when the flow becomes emergent. So this is exactly what has been termed of late as living fluids. You can have living fluids in different kinds of systems. You can have, let's say, active pneumatics. You can have microtubule kinesin mixtures. The one we are looking at is bacteria. But the governing principle is the same. You have a dense suspension of microsomas that self-propel, and they can organize together. So these are the systems that we'll be considering. And just to mention, I mean, over the past about 10 years, there's been a lot of interest in this topic. A lot of papers started coming out, and this is really just the tip of the iceberg. But what I want to point out is that many of them, they mention either mesoscale turbulence or active turbulence, bacterial turbulence, and so on. And this brings us to our motivation as people who work on inertial turbulence. We wondered what is alike between active and inertial turbulence, apart from, of course, a visual similarity, because they're both chaotic, they're both vortical, but does it go beyond a visual similarity? So how do we approach this question? First, we started, of course, with simulations. So you can, for instance, there are different models. The one we work with is a continuum model for bacterial turbulence. And again, what you see in the first movie is an experiment. And to the right of it, you see movies from simulations. This is the governing equation, which is essentially the Navier-Stokes equation with additional terms for forcing in Fourier space and in physical space. This is the last one is called the Toner-2 term. You don't need to focus on the different terms of the equation. The only important parameter that you can note is that there's an injection of energy with this alpha. So when alpha is negative due to this sign here, it injects energy into the flow. And I do want to mention what this equation actually simulates, because see, that is again a system of many, many microswimmers and a little bit of solvent fluid. But if you coarse grain that image over, let's say, tiny volumes, what you'll have is the collective motion of thousands of bacterial cells. So this velocity essentially represents an averaged velocity for a tiny fluid volume over there. So this becomes a continuum version of the sort of discrete system over there. And it's also incompressible and so on. So we wanted to study the properties of these flows, and you can see that it changes. I mean, the flow visually changes again, whether you are in a mildly turbulent regime or a highly turbulent regime. But then again, is this really like turbulence? And people had asked this question before, of course, for active turbulence. And what they found was that active turbulence is, for instance, not universal. Inertial turbulence, we know, has an energy spectrum scaling, which is universal. It doesn't depend on the large scale forcing and so on. But active turbulence, it turns out, was, the energy spectrum scalings, it depended on the level of activity. So people concluded not universal. Similarly, they also concluded it's not intermittent. So if you look at quantities like velocity differences, you look at the distributions, everything is very quiescent and Gaussian. So people felt that, well, active turbulence is basically a chaotic flow, but it's not really universal or intermittent. So it's not really like turbulence. So we wanted to revisit this question and see if there is something more to it than this. So to cut a short story even shorter, what we found is there's an emergent universality. So again, we just look at the energy spectrum scaling for increasing levels of activity. So this previous study, of course, was not wrong. What they were looking at was just a small part of this sort of entire phase diagram of delta, where delta is the scaling of the energy spectrum. What we find is that beyond that regime of non-universality, there is a sort of a phase transition to a universal spectral scaling exponent, which you can also theoretically derive using the energy spectrum equation, certain assumptions on a few of the terms and so on. And you arrive at a constant slope of -3/2. Interestingly, the flow is also actually intermittent in this state. So there is a critical alpha C again, which is activity. When you move the system beyond that activity, if you look at this quantity again, which is the velocity difference projected along a separation vector, you look at the distributions of those and you see how far they are from Gaussianity by looking at the flatness of the distributions, you again see a phase transition. Well, sort of a transition at least at an alpha C. So we found that active turbulence is in fact universal and intermittent. So to close the picture, we thought let's also look at the nature of chaos, because together with intermittency and universality, chaos really determines inertial turbulence. And it's also very important from a biological perspective because these systems need mixing of nutrients, limited resources, and so on. So how you measure chaos, I mean, the next talk will speak a lot about OTOX and so on, so you'll see it in much more detail. Here we simply do a twin simulation where you have identical simulations, you perturb one of the twins, and you measure the difference between the simulations and see how they evolve. So here the perturbation was just a small Gaussian spot. So you see that spot grows, dominates the field. And you can average these kind of measures like the different velocity or vorticity, you get a decorrelator. And if you look at these averaged quantities over time, you get a very typical chaotic growth curve. So you have an exponential growth phase followed by saturation. You can fit lines, get Lyapunov exponents, and so on. And what we found is that these systems are maximally chaotic beyond a critical activity. What is interesting, though, is that the nature of chaos also changes. When you go from mildly to highly active turbulence, I won't go into the details here, but highly active turbulence sort of acquires a scale-dependent Lyapunov exponent. So you get really first the saturation of small scales, which are the large wave numbers, then the larger ones, and so on. So there's a hierarchy of saturation of scales. So that shows that there's a whole spectrum of Lyapunov exponents. And this is, again, similar to inertial turbulence. So now I could have come to a summary of my talk and said, well, what we found is that active turbulence has an asymptotic state beyond a critical activity, where it becomes universal and intermittent, multi-scale chaos and everything. But you see, there's a however. And in my limited experience, everything in turbulence has a however. So what is the however here? That's what we find in the Lagrangian picture. I mean, so if you consider the Lagrangian aspects of these flows, you see some differences, and this might be a bit of a repetition for some of you. I've presented this work before, but just to remind you, what we find is for increasing values of activity as the flow becomes more and more turbulent, if you look at the diffusion of tracers, then you have at low activity, which is on the left, you have your ballistic to diffusive transitions. But at high levels of activity, you get a very robust ballistic to super diffusive transition. So there's anomalous diffusion in the system. And if you do the statistics of individual trajectories very carefully, you can show that this anomalous diffusion actually can be traced back to what are known as Levy walks. They are these specialized kind of random walks which give anomalous diffusion. And associated with, I mean, if you have super diffusion, any other Lagrangian measurement will also be colored by that aspect. So if you look at, for instance, first passage statistics, there's a clear distinction between persistent trajectories and diffusive trajectories. What we also found was that the nature of Lagrangian trajectories, or actually their future, it depends on where you start in the flow. So all of this anomalous diffusion can again be traced back to the Eulerian field in some sense. So what I'm trying to tell you with these two slides is that there are also differences between active and inertial turbulence, which are most prominent in the Lagrangian framework. So that brings me to my final summary. So I can say that active turbulence is universal and intermittent. It has maximal chaos. So in some sense, our work shows that it brings it back in some way closer to inertial turbulence. However, it seems that some bacteria, they live life in the fast lane. So the Lagrangian pictures of active and inertial turbulence are quite apart from each other. So I thank you for your attention. I also want to end by thanking my collaborators. So I worked on this with Rahul, who's now in OIST in Japan, Martin, who's in Geneva, and Somriddhi, and our collaborators at NCBS, Suresh and Shashi. We are doing some experiments in their lab, basically, again, looking at this active turbulence system, but with the addition of polymers and so on, to see whether we can tune these exponents that we get of anomalous diffusion. And here are some references for this work. So yeah, I thank you for your attention.
Thank you, Siddharth, for the wonderful talk. Any questions from the audience?
Hi. Hi.
Manas has it.
Yeah. So you had a plot in which you plotted Lyapunov exponent as a function of alpha. So the Lyapunov exponent remains the same for some up to some critical activity and then drops. Is it?
The opposite. It increases up to a certain activity and then it's not changing anymore.
So I see. So that is
yeah. So it increases and then it stops because This the left side direction is increasing.
okay. So,
Yeah,
so okay.
yeah.
And did you also calculate things like butterfly velocity and?
You mean the spread velocity of these perturbations?
Yeah.
No, we didn't. I didn't look at that.
Okay.
That might be interesting to see. Yeah.
Okay. Thanks.
Yeah.
This is regarding this difference between the two different kind of turbulence. Is it known whether, this, if you calculate velocity, there are actually correlations. The exponents there are similar to inertial turbulence or?
For velocity correlations.
Yeah.
I don't. Well, structure function. We did look at structure function exponents, but they weren't similar to. They weren't similar to inertial turbulence. And I think you can get to velocity correlations from that. So they are different, I think.
So even in the regime where you have intermittency, the intermittency is very different. Is that the lesson to take?
Yes, because also we don't have very extreme intermittency in this case, which you can get in. I mean, in inertial you can get as extreme as you want, but here we don't have that.
Okay. Maybe just one more question. It's just so when we talk about active turbulence, does it matter what kind of model you use for active matter? Are there many different kinds of active turbulence?
Yes. So the honest answer to that is yes, because there are many different models and people have found different kinds of transitions and so on. But what we are at least interestingly seeing is that the asymptotic state sort of exponents, they seem similar across models, but there hasn't been an intermodal comparison yet. But we've tried to fish out in papers and we find a similar exponent. So yeah, but the other details of the models change, and that's why the flow states you can get can also change.
Okay, thanks.
See, you were talking about this dynamic heterogeneity. Basically, in the context of glass, it has got some specific meaning.
Yeah.
So one of the understanding of that is that when you have the coexistence of slow and fast regions in the system.
Yeah, it's exactly that.
But these are vorticities.
So this is a vorticity field, the image. Now what I'm showing you is exactly if I start my tracers in that region, you see the vorticity patterns look different. They are more streak-like. Whereas if you look here, you have these round spotted vortices. So if I start my trajectories there, they mostly go in a persistent manner, ballistically, basically. Whereas if I start over there, they go diffusively. So if you look at MSDs here, this would go ballistic, this would be diffusive. And there's, of course, every trajectory will go through both regions, and that's why you get anomalous diffusion, something between an exponent of 1 and 2.
You have a spatially dependent
Yes.
dynamics.
And in fact, if you look at where these are located in space, so these red spots are showing in one snapshot where these are, so that there's also a spatial concentration. So there is, that's why we called it dynamical heterogeneity, because it has that similarity with what you mentioned from glassy systems.
I guess this is why you have a peak at 0 for GFR. These are not the finite size particles.
This is the cluster of tracer particles.
No, GFR, the peak at R is equal to 0.
Yeah. That's because these are highly concentrated, right? That's right.
So they don't have any size, finite size.
No, these are tracers. So there's no finite size. Yeah. It's just cluster.
Thanks.
We can take one more question.
Look at, Karman-Horvath equation, like for this equation, for this governing equations. Like Karman-Horvath equation is the one that governs the correlations, velocity correlation.
I haven't looked at that.
Okay.
Okay, so let's thank Siddhartha again for the talk.